be working now, hopefully. Can anyone let me know if you can hear me?
Hear you?
Yep, you can hear me?
Yeah.
Okay, so Kraken should now have posted the YouTube link. Brilliant. So perfect timing. Sorry about some of the technical issues. I've joined on my iPad for the first time, and not only could I not turn off notifications, I also couldn't speak with the microphone. Anyway, everything is operating smoothly. So first of all, I'll run through just some housekeeping rules. Make sure you keep your microphone off for the duration of the talk. We will take questions at the very end. We're going to have two lovely speakers, but please save your questions until the very end. You can write them in the chat throughout the talk. I will keep a list of them, and then I'll ask them at the end. So without further ado, I will introduce our lovely speakers and let them get going. We have Anais from Microbites and Charbert from Microbites. It's going to be in two halves, and they are both PhD students on topics around the microbiome, and that's what they're going to be talking about today. And so we have two real experts. We're so blessed to be joined by them. And without further ado, I believe, Anais, you are starting, so you can feel free to...
Yes, can you hear me okay?
Yeah, I can hear you. It's a little bit of background noise. I don't know if that's for everyone else, but yeah, I can hear you.
Okay. I'm just going to share my screen and then I'll get started. Okay. Is it working? Okay. So today we're joining Biocord for a talk about microbiomes. So this talk is a small contribution to the World Microbiome Day that is happening on the 27th of June. And we just wanted to discuss a bit about microbiomes as a whole, actually. So first of all, we wanted to give a shout out to the lecture that happened last week with Microbiomes. So we are also collaborating with Tess. And if you want to know a bit more about microbes in general and how they impact our lives every day, I suggest you also listen to that talk. It was a great introduction to microbes in general. So how microbes impact our lives, but also the environment and medicine, and also agriculture. So what are microbiomes? Well, actually, microbiome refers to microorganisms, but also their environment. So all the microorganisms, including bacteria, but also viruses or yeasts, and the environment itself. So if we talk about the human microbiome, it doesn't mean much. We should actually talk about the human gut microbiome or the human lung microbiome or the skin. But there's also the animal microbiome or the food microbiome, as well as the soil and ocean. So Charlotte and I are both PhD students on topics related to the human gut microbiome. So let's start with this one. This microbiome, we've heard a lot about it. It's quite often on the news because so many nice research are being done right now about it. And we're discovering a lot on its implications. So we know it helps us develop our immune system, and it also helps us digest our food. And when everything is fine, it's highly beneficial for us. But when something is going wrong in the gut microbiome, we see that it's actually related to some diseases that can be gastrointestinal diseases, such as irritable bowel disease, IBD, or also linked to more neurological diseases. So it seems to be implicated in many different aspects of our health. But there's not just bacteria in the human gut microbiome, there's also a lot of viruses and fungi that are a lot less studied right now. So what Charlotte and I are actually working on is trying to understand a bit better how those microbes work together and how they interact. And to do this, basically we use microbes that have been isolated from stool samples, and we put them in those type of bioreactors to study their interaction in vitro. And hopefully they live nicely together and have a chill afternoon in our bioreactors, and that's what we actually look at. But as I mentioned before, microbiomes are everywhere, so we've looked at the human microbiome a bit, but it has also a very important role in the ocean and rivers, for plants and soil, and also for so let me introduce you a bit to what we do at Microbites. So actually we're taking microbiology research papers, and we breaking them down into little bite articles. So we simplify the articles and make them a bit more digestible to read. And as you can see in the video, we try to cover a lot of different microbiology topics. So we've looked at, for example, those two bacteria that are cooperating in their environment, but we try to cover topics from viruses to fungi, and try to be as broad as possible on the topics. Because our team is from different countries and we all speak different languages, we started the website in English, Dutch, and French, and more recently we also have the website in Spanish. And by next week, Saturday, we'll also have it in Russian, hopefully. And on Microbites, we've actually looked at several papers related to microbiomes. So here you see a couple of papers about the human microbiome, but also the animal microbiome, as well as the food microbiome, and one of the most recent one on soil microbiome. So if you're interested in stories about microbiomes, you can read them on Microbites later. And actually today what we wanted to do is to discuss two examples of microbiomes, so two research papers that we've done on Microbites, and talk to you about it, how the research was done, and different aspects of microbiome research. So it won't be gut related tonight, so we're a bit out of our own expertise, but actually microbes and microbiomes are fascinating in all their way, so we wanted to share that with you. So I will start with the sourdough bread, and then Charlotte will talk to you about the ant microbiome. So what is actually sourdough? Well, it's a mix of flour and water and bacteria that are in the environment, but also contained in flour and water. And this gives you an initial starter, and if you feed it regularly, so basically what you do is take a bit of this initial starter and add more water and flour, and then every few days, like on a regular basis, you do this, and at some point you'll have what they call active starter here in the paper. And this active starter, actually, you see it. It's bubbly. It smells like sourdough bread. It smells very nice, actually. You see it's alive but healthy. When it starts smelling a bit weird or the color is not right, then you know it's contaminated, and you can put it in a bin. And when it's nice and healthy, you can use this active starter to actually make your bread, and your bread will be nice and with this nice sourdough flavor. So what is actually in sourdough bread? Well, it's a mix of different microbes, so some yeast, but also some bacteria, mostly lactic acid and acetic acid bacteria. And this community of bacteria, this microbiome, will create different products. So first, organic acids, such as lactic acid and acetic acid, but also, of course, CO2 that will make the bread rise. So in a recent paper a few months ago, Landis and colleagues looked at the diversity and the function of sourdough starters microbiomes. And this was a very interesting study because it was actually a citizen science project. So they gathered samples from all over the world, and it was the first study actually that did that. There's a lot of research going on about sourdough bread. It's mostly done in Europe or in specific locations. Where here they really studied samples from, mostly from the US, but also from Europe and Thailand, but also Australia and New Zealand. So overall 500 samples. And they also tried to gather a bit of data about the samples, so how they were stored, but also the feeding frequency. So I told you can feed them regularly, but actually, depending if you put it in the fridge or at room temperature, you won't have the same feeding patterns. Also, the age of the starter and its origin. So if it came from an individual or a business. And the first thing they looked at is the community profile of those microbiomes, all those communities. And as you can see in the profile, it's highly dominated by one specific microbe. So at the bottom you have the yeast, which is almost all the time Saccharomyces cerevisiae. And actually that yeast you should know very well because it's also the one that gives us beer. And at the top you see the different bacteria, and here you have a bit more diversity, but if you look at each sample, you will see that it's mostly dominated by one bacteria. So here the pink one, and here, for example, the red is mostly dominating those communities here. But what they found is actually an underreported type of bacteria, and most studies before they didn't report those that much. So what they found is that acetic acid bacteria were actually quite prevalent. They were found a lot in the different communities, but at very low abundance. So those acetic acid bacteria, it's those ones here in yellow. So you see them here and there, but quite low abundance compared to other ones. Then I tried to see if specific microbes would be enriched in some specific conditions. So they looked at a total of 33 parameters, so including geographic differences and age and storage location, to see if it could explain community composition. But actually all those parameters only explain 10%, which is not that much. But they could still see trends. So for example, they saw that in younger starters, you would have Lactobacillus plantarum and brevis that were more enriched in those younger starters, while in older starters, you would more have Lactobacillus sanfranciscensis, for example, or the parvovirus. Then they try to sort of understand how those microbes were interacting in those communities, and to do that they tried to recreate those interactions in the lab. So what they did is to do co-cultures, so put two microbes together, so either yeast or bacteria or a combination, and then they recreated the sourdough way of making. So they were transferring the culture every other day, like you would for sourdough starters. And then they looked at the composition of those co-cultures after a number of transfers. And basically they were just looking if, for example here, if just one was taking over this small community, or if they were actually coexisting, like here. And what they actually found is that the interactions they were seeing in the lab were actually quite representative of the interactions they saw in the sourdough samples. So here on the left, you see the co-occurrence pattern. So basically, if two bacteria were quite often be found together, then we would have a blue line between the two. So those two guys, for example, were commonly found in sourdough samples, while those two connected by a red line would be negatively co-occurring. So that means that usually they were not found together in the communities. That's what's happening in the sourdough samples, and that's what they saw from their co-culture experiment in the lab. And as you can see, most of the lines are actually the same, the ones with the star, so the ones that actually had that were the same than here, except then that one here that is now blue, but here it was red. So that means that in the lab they could coexist nicely, but they were now not found together in the sourdough sample. So that could be explained by a number of reasons. And then they looked at the different communities. So that's a little video that you will see if it's moving. Yes, it's moving. You will see that the different communities in the tubes will actually be rising. So the dough will be rising at different rates and at different heights depending on the community. So it's really related to the community composition and how much the dough will rise and how fast. So that was quite a nice observations that they made. Then they looked at the different chemical signatures from all the communities. So here you have all the chemicals they looked at, and basically they found 14 dominant sensory notes, from yeasty to green apple and solventy, but so many different kinds between all the communities that they had. But actually the differences between the communities were driven by the abundance of those acetic acid bacteria. So those acetic acid bacteria, remember, they were found in a lot of different samples, but at very low abundance. So it seemed that those guys, although they were underrepresented in the community, they seem to have actually a big influence in the aromas found between the different samples. Okay, so that was their paper. So basically, what they found is that different communities in the different sourdoughs were enriched by specific microbes in different conditions. But these communities will actually be impacting how much the dough is rising and how fast, but also the aroma of the sourdough bread at the end. And they also found that those acetic acid bacteria are actually very important members in the community. So that was also a nice result from this paper. So if you're interested in other food microbe stories on Microbites, we also did a cheese paper. And as a French woman, I'm very grateful for the microbes to give us those nice fermented foods like bread, cheese, and wine. So don't forget that microbes are everywhere, and including our food. So from this, I will give the floor to Charlotte, and she will talk to you about the ant microbiome. Let's stop my screen.
Thank you, Anais, for the nice introduction and the sourdough bread story. I hope you can hear me. So what I wanted to do is talk about another different microbiome, namely that of the ants. So we've seen in the sourdough story that we can look at a microbiome as in what is in there and what is the function of the different components of this microbiome. And here with the ants, I wanted to show, okay, we can see what's in there, but how can it be influenced? So the ant microbiome We have nice stories already about ants and microorganisms on the internet, so I looked up a few. And actually, ants can use microorganisms to their own, for their own. So they can grow fungi in their own hive, and then they will eat this fungus and then use it as a food source. But the other way around is microorganisms using the ants as a food source. So you've probably seen this already on the internet if you've been...
Sorry, can I stop you just for 5 seconds? I don't know if people can see your slides.
Oh, okay. Sorry.
That's all right, don't worry. It works when we practice, I can say, everyone.
I should do my entire screen. Let's see.
So I can see your screen.
Okay. Let's see if it works now.
There you go.
You
That's
see it?
working for me now. Yeah.
All right.
That's working.
Sorry for
You
that.
can- Sorry for the
Okay, you didn't miss much. It's just these two pictures. So as I said, I will put in a pointer. There you go. Okay, so in this picture, you can see that the ants are using the fungus to grow in their own hive, and then they eat the fungus and feed it to their offspring, which is really nice. And then a more cruel story is these microorganisms that are using ants. So they infect the ants, and then they will use the nutrients of the ants, so to speak, to grow on their own. So if you Google on ant and zombie, you will find a lot of these cool stories of microorganisms doing this. Also, a bit closer to the topic that I will be talking about is that the gut bacteria in these ants can be very important. So it is shown in all living species that the gut microbiota, they can break down complex nutrients which normally cannot be taken up by the body of the animal itself, including the humans. And this nice story about ants, which came out quite recently in January this year, they show that the gut microbiota are really necessary for the growth of the panzer of this Florida turtle ant. So without these bacteria, the panzer was less strong, and then the bacteria would be less protected against things from the outside, which is really nice. So how do ants and, well, most animals get the bacteria? So one thing is obviously the food we eat. And for some ants and other species like bees and termites, they do trophallaxis, which is some kind of mouth-to-mouth feeding. Which means that one ant eats and then it can regurgitate the food to another ant, which will swallow it, and then it will go to their intestines. You can imagine that if you eat something that's spoiled, then there's a lot of bad bacteria and sometimes even fungi on the food that can infect you. And the researchers in the article that I want to present, they wanted to know, like, how do ants control for these harmful bacteria in their food. So this is the research paper that I wanted to talk about for a bit, in a more simple version than that they present. Is how do these ants make sure that the good guys will go through the stomach and enter the intestines, and that the bad species, they will die before making the ants sick. So in particular, they looked at this Florida carpenter ant, which is called a formicine ant. Why is it a formicine ant? These ants have this specific acidophore in their bodies, and it produces a very acidic toxin, and it is made mostly out of this formic acid. And formic acid, as the name says, it's named after the species where they found it. So the formicine ants. And I don't know if there are Dutch speakers here, but I'm Dutch, so in Dutch it would be called mierenzuur, which, if you translate it literally, it's called ant acid. And so these ants, they use this formic acid, or actually the whole toxin, they suck it up here from this acidopore into their mouths, and then they can spray it again to protect against predators, because it's an acid. But what the researchers wanted to know is, okay, the ants can put it in their mouth and spit it out, but is this poison also swallowed? And if it is, does it allow for microbial control? So can it limit the pathogen transmission through mouth-to-mouth feeding? And also, do beneficial bacteria, can they go through this acidic environment? And I wanted to show you some results. So first they looked at the pH of the stomach of the ant. Right before feeding, or 4 hours after feeding, the pH was quite high, which means it's not very acidic yet. And then after they fed, you can see that the pH goes down, so there's more acid in their stomachs. And then when they gave some feed again, you see that the pH, it goes up again. So they fed the ants with sugar water, which should make the pH go up again. Then what they wanted to look at is, okay, if we cut off the ants from the ability to suck out this poison from this poison gland in their body, is the pH still going down? So here you see that if the ants were able to suck up the poison, you can see that the pH goes down in their stomach. But if the ants cannot suck up the poison from their poison gland, then the pH stays relatively the same. So it's really this poison, this formic acid and the other components that make the pH go down in the stomach of the ants. And then they wanted to see, okay, we looked at this Florida carpenter ant. Does this happen in other kinds of this formicine ants too? And yes, you can see that in almost all cases, the pH is lower when they have access to their poison glands compared to the ones that don't. Okay, so they now established that the pH is going down because of this poison gland. And here they wanted to show that bacteria cannot survive if this is happening. So what they did is they fed the ants sugar water, and in this sugar water there was this bacteria that is very pathogenic to ants, so it kills the ants. And then they looked at, okay, these ants that have access to this poison, does it actually kill this harmful bacteria? And here you can see they looked at the amount of bacteria that was still alive in the different compartments of the digestive system. So they looked at the bacteria in what they call the stomach, so it's a crop. And they also looked at the bacteria alive in the hindgut, so you would compare it to our small intestine. And what they saw that after 4 hours of feeding, so they fed at 0 hours, and 4 hours later you can see that there are almost no bacteria of this kind alive anymore in the ant. And they also, after 24 and 48 hours, there's still no live bacteria. They also looked at if the bacteria gets transferred to this hindgut, so the small intestine, are there any live bacteria in there anymore? And indeed they saw that after 0 hours of feeding, there were some bacteria that went through here immediately, so they were not very long in this acid environment. But as time keeps going on, you can see that even there the bacteria were not growing anymore, or they weren't even there in the first place. They're all dead. So here they show that this acidopore grooming, as it's called, like sucking up this acid from their gland, it can really control growth of, for at least this specific microbe, in the ant. As said, these ants, they can give their food to the other ants, which means that if they do this acidopore grooming, they get this poison from their glands into their mouths. This poison will also get transferred to the receiver ants. So they did this nice test where they had the donor ants that were fed with this pathogenic bacteria, and they made them live together with these receiver ants. And these receiver ants, they did not have the capacity to do this acidopore grooming, so they did not have access to their acidic poison. Then in another group, they had these donor ants that were fed with this bacteria, and they also do not have the ability to eat their poison. And they grew them together with these receiver ants, also no ability to get their stomach very acid. And what they saw is when this donor ant did have access to their pores, you can see that their survival rate is higher than all the other groups. And because this pathogen, it's once you have a few of these bacteria in your ant, they almost certainly die. And then we see the receiver ants of this group. They have a higher proportion survived after 12 days of ingestion of these killer bacteria. And we can see very nicely that this donor ant and the receiver ant group that did not have access to their poison, they were not able to kill the pathogen fast enough, or not at all. And you see that, only a fraction of this group survived. Not significantly from each other, but they were significantly different from the other two groups. So here they show that this mouth-to-mouth feeding, this trophallaxis, can limit the pathogen transmission in these ants, which I think is a really cool experiment. And then what they were thinking is like, okay, it kills bacteria, certain bacteria, but what if it kills all the bacteria? Are there any bacteria that can go through this, let's say, chemical filter and survive? So they did the same experiment. They gave the ants a bacteria. This time, it was a bacteria from the Asaya family. And this bacteria is found in a lot of, in the microbiome of a lot of ant species. So not only this carpenter ant, but also other ant species. And they were wondering, okay, if we do the same tests again, can we see if this species can withstand the acidic environment in the ants? And here you can see nicely after time point 0, so just after the feeding, obviously the bacteria are there. And you can see that after even 4 hours, the bacteria are still found alive, although not in as high as a quantity as before. But if we look at the previous slides where I showed you the pathogenic bacteria, all the bacteria were dead at this time point. So this Asaya species bacteria were able to withstand the very acidic environment in the ant gut. And then if we look at the intestines, and here in the intestines is where most of the resources, the metabolic compounds, are taken up, we can see after 0 hours of feed and half an hour later, these bacteria are not there yet, which could probably mean that the bacteria did not go from the stomach yet to the small intestine. But after 4 hours, you can already see the numbers going up a bit, and after 24 hours and 48 hours later, these bacteria are happily thriving in the gut, gut of these ants, which means that the formic acid doesn't kill this bacteria, species specific, and can kill other bacteria, which means that there's a filter function of this formic acid, which I think is really cool. So the conclusion of the paper is that this acid, so this poison gland grooming, can kill harmful bacteria. It can prevent disease from spreading, as they showed in the donor-receiver pair experiments. And the last thing I showed is that they can act as a filter for these potential beneficial bacteria. They talked about, in the paper, they talked about further research. So are there other things that influence the acidity of the crops, so the stomach? Is this particular done in this specific ant species, or are there other species of ants that have a similar mechanism? And also, is there an optimal secretion of this poison gland? Is there an optimal compound how do you say that? Mixture that is better or worse to kill specific bacteria. So instead of showing you guys a microbiome, I wanted to show you that in order to understand the microbiome, we can also look at the organism and see how can an organism influence their own microbiome. And I thought this was a really cool story. So, as Anaïs said, you can join us on World Microbiome Day on the 27th of June. We have a nice collaboration with Microbiomes, which is a podcast that we'll be doing, which is the also from Tess, the girl that presented last week. What we will be doing, we will gather a lot of questions about microbiomes, and we will try to answer them on our website. So if you have a question about microbiomes that you want answered, but you can't think of it today, or it's not answered today, you can submit it to our website via Facebook or via Twitter or the website itself. And we will post the top questions and the answers on our website on Microbiome Day. Also, we're looking always to find people to help us in doing everything we do. So if you want to join us in writing articles for the website, you're always welcome to. If you would like to translate it to a foreign language other than English, you're very welcome to do that. At the moment, we're looking for Russian translators. And we are also in need for some Dutch and French translators. If you want to do that, just go to our website, click on volunteering, and then most of the time we answer on the same day, if not one day later. And that's it. I want to thank you all for your attention, and we'll be happy to take some questions.
Brilliant. Thanks so much. That's such a fascinating talk. And thank you to both of you for coming here and talking to us. The chat's been very active, so everyone's been very excited. So I've got a list of questions. So if anyone has any more questions, please do write them in the chat now and I will get to them. So I've taken them in chronological order. So I think Anais has some questions first. So the first question is, in the sourdough, what is responsible for the starter to change their microbiome composition?
Yeah, so maybe I wasn't very clear at the beginning, but actually the microbes come from the environment, but also from the water and flour that you put in. So there's always microbes everywhere. And basically, if I do it in my kitchen here in Belgium, the starter will probably be different than if you do it in your kitchen wherever you are. So that's where they come from, the environment.
Okay, great. Sorry, my microphone is working. So the next question is, which microbes make the best sourdough?
I don't think they've answered that question in that paper. I guess because different starters will give you different flavors, then you would have to try them for yourself. But again, because they come from the environment, you're kind of stuck with the ones you have and the community you have in your own sourdough. So you could try it from different bakeries, or like if you have different people doing sourdough bread around you. But if you do it yourself, you're kind of stuck with the microbes around you, I guess.
Thanks for answering. So the next question is there a correlation between bacterial species and the location of a starter? So for example, do German sauce starters have a more I'll pronounce that, bacter species compared to the Irish-based starter?
They didn't really see specific signs of that about location. Maybe it wasn't significant enough because they didn't have enough samples from specific locations. So again, they had a lot from the US, but not a lot from Germany. And I don't know if within the US they had enough in the same locations that they could do actual statistics to see if they were that different. So I don't think they saw anything popping up about location. No, more about if it was a young or old starter, and also the type of flour that you will put in. So the type of wheat in the flour. That they saw some signs and differences in the communities, but not really about location.
Yeah, that's interesting, because I probably would have expected there to just be random differences just by geographical location, but it's interesting that there's not. So anyway, next question.
Yeah.
It was, I wonder if we eat ants, will we inherit their gut microbiome?
That's a nice question. So the amount of bacteria in this ant microbiome would be so small, probably they would be overgrown by all the other bacteria that we eat, and there's also already bacteria in our guts, and probably they are more adapted to our environment, which makes them very competitive for our guts. So probably no, because they will be outgrown by all the other bacteria in our guts.
Right, yeah, it's kind of like the C. diff thing when you take antibiotics and you allow C. diff to be able to overtake. Let me just get the next question. The next question was, how do you extract bacteria from ant stomachs in the first place?
Yes, so I would imagine, and I don't know it by head, but I think you'd have to open the bacteria, open the ants up, and then with a little swab, take out the bacteria and streak them on an agar plate, so you can count the colonies of the bacteria. So actually you'd have to kill the ants in order to take out their contents.
Right, yeah, so maybe like a squash prep or something like that, or just a dissection.
Yes.
The next question is, how do you farm the ants?
Okay, so should have looked at it. These ants were probably grown in an ant farm. I'm just looking at the material methods of the paper because I didn't really look at it. So I'm so sorry. So they coated plastic containers with plaster ground, and they had a constant temperature of 25 degrees. So they were just in containers with some plaster on the ground. And then they will just fed the ants with water and honey water 2 times a week. So just in plastic containers with some little bottom to walk on.
Right.
So it's not from the ants giving a heel or anything.
Yeah. So I also work with insects, so they can kind of, you can keep them happy with very, very simple things. They're not like mice, right? They just give them some space and some food, and they're all right.
Yeah.
Next question: are all non-pathogenic bacteria resistant to formic acid?
No, of course not. There's like this interchange between the bacteria and actually the organism that they're growing in, because these bacteria, they constantly evolve, and if such a change is good for the bacteria, they will keep on growing in these harmful environments. So there must be pathogenic bacteria that can withstand this formic acid. But on the other hand, they are in such low quantities at the moment that they might be overtaken by the rest of the microbiome, or they're not adapted to survive after this acidic environment further in the guts of the ant. And of course, also good bacteria will be killed. Bacteria that could potentially be beneficial for the ant, that are not adapted to this acid, they will also be killed. So it's a bit of a trade-off.
Right, yeah, that makes sense. So the next question I've got is, if the starter microbiome changes with water, how would you test the starters, control for it, sorry. So do they use distilled water?
So they actually sequenced directly the starters that they received. So they didn't grow them in where they were. So I guess different people had different waters and different flour in different environments. So they didn't grow them per se. Yeah, they just sequenced them when they received them. The only thing they did in the lab were the co-cultures, so that's completely different.
Oh, that makes sense. That makes sense. Yeah, thanks for that. So the next question, what kind of controls do you have in place when testing changes in the microbiome if it's dependent on the flour and water? So a similar kind of question.
Yeah, so again, if they wanted to test that, they would have had to have different types of water from different places, I guess. I'm not sure that the water is really the main thing in this case. I think the bacteria from the flour and the environment, and also the type of flour that you feed them will influence it probably more than the water itself. But yeah, for sure, if they would want to test it in the lab, they would need different types of each of the ingredients, which is just the environment, flour, and water. That makes sense.
Yeah, that makes a lot of sense. Yeah. So I've got I'll probably take two final questions because I don't want to keep you here all day. So penultimate question. So you mentioned the amount of acid being too small to be extracted from ants. Can we engineer the genes in bacteria to get a larger amount?
I think I said that the amount of bacteria was too small to be transferred. I'm not sure if I said that the acid was too small to be transferred. So I don't really understand the question. Let me read it.
Whoever asked that question, I can go back. Do you want to maybe rephrase it a little bit? So whilst they are rephrasing, I'll give you the last question. It says, can we engineer the bacteria to be resistant, or would that result in disaster because of horizontal gene transfer?
We could try to engineer these good bacteria indeed, but as you said yourself, there's a chance that these genes will hop over to other species. And then, if once it's in the environment, it could be transferred to all the bacteria that come in contact with each other. But in any case, there would be a lot of different mechanisms to withstand the acid. I don't think they would do it in nature because it's not useful for us, I guess.
Right, yeah, I understand perfectly. So that's not rephrased it. So if there's no last minute questions, I want to say a huge thank you to both of you. There's been so many thank yous in the chat if you've been checking it in the Lectures and Conferences channel. It was so interesting. We've absolutely loved hosting you. And yeah, I'll put the link to your website in the channel as well, so you guys can go and check out Microbites. It's such a cool organization. What you do is amazing. You know, helping to bring the microbiome and microbiology out to people, and even translating. You know, that's such an important part of science communication. And you guys are awesome. So yeah, without further ado, the recording will hopefully be available later. And thanks for joining us. We'll see you again next time.